I'm here today with Stefanie Countryman, my colleague who works at BioServe Space Technologies. Stefanie, you want to tell a little bit about what you do at BioServe?
I work with BioServe Space Technologies at the University of Colorado, and we fly life science experiments in space on the space shuttle, and now that the space shuttle is retired, we fly them to the International Space Station. So my job, I run a program where we try to keep different types of organisms alive in a space environment, and we try to get them to act as naturally as possible in an unnatural environment.
In 2008, BioServe wanted to send spiders up to the International Space Station. So Stefanie contacted me and asked me if I could give some advice on how to design the habitats that were going to be used. So you brought some with you today.
I brought some of our first designs that we had that actually flew our first experiment. And as you know, the big thing was we wanted to take video of the spider, so I needed your advice on how we could keep the spider in the view of the video, because as you know, the spiders sure like to hide in places where videos can't see them. And then we talked about what was the necessity for a spider. And I believe you told me the biggest thing was water.
Yeah.
We ended up with 2 spiders in the primary section of the habitat on station, and so then we were concerned that the 2 spiders wouldn't spin their webs because it was such a small space and that they would kind of infringe upon each other. But the exciting thing was we actually had them both spin webs, and they spun them side by side right next to each other.
We've worked on 2 different spider experiments in space. So one was orb weavers that were sent in a habitat kind of like this. And then the other experiment, we sent jumping spiders in a habitat that looked like this. That was proposed by a kid, right?
From Egypt.
Mohammed from Egypt proposed sending jumping spiders up to space to see if they could learn how to catch their prey without jumping, because they tried to jump and they'd go flying off. And they did. They managed to capture their prey just fine. And I thought one of the fun things about this experiment was that the astronaut who got so excited about it,
Sunny Williams.
We have some great images of her from NASA checking the experiments. I just thought it was kind of cool that it was a female astronaut who was really excited about the experiment.
The 2 spiders actually lived for 100 days, and when she was packing them up, she said she was more nervous for their return to Earth alive than she was for herself when she comes back in the Soyuz. But they were both alive when she sent them back. And
that's great.
And it was pretty exciting.
At the museum, we have a really great little exhibit on the spider experiments, spiders in space, and included in that exhibit is a little bon voyage note that was signed by the 3 astronauts that were on the International Space Station. I thought that was really fun. That was her initiating that little note.
All of that.
What other kinds of animals have you sent up? Or organisms, I should say, because haven't you worked on some bacterial experiments?
We have flown all kinds of things. We've flown the fruit flies for the spiders. We've flown a lot of different types of bacteria, Salmonella, MRSA. We've flown different types of viruses, C. elegans, which is a small little nematode.
Sure.
We've flown crystals, and we've done butterflies in space. We've done fish. We did Medaka fish.
What's your biggest challenge when you're designing one of these experiments? I mean, is the biggest challenge to design the habitat or to just figure out the biology of the animal?
It's both. It's really both, because we have to understand, in the Space Flight Center, we have to understand the biology of the habitat, of the organism, in order to design a habitat that will support that biology. We weren't able to keep them alive just in a cage that was fairly large, that had airflow, and so I couldn't keep them alive there. And if I can't keep them alive there, I'm certainly not going to keep them alive in a small container that has limited airflow. So, so the first thing when we do this is really to understand the biology of the organism, how they live, how they interact with each other, what kind of foods they eat, and that's where your expertise comes into play for that. And then we can design a habitat based around that, and all of that's based upon the safety requirements of NASA, because in NASA nothing can escape from the habitat. There has to be what they call levels of containment. And so everything has to be contained, and depending upon the organism and how dangerous it may be to an astronaut, it has to be contained in 1 level or 2 levels or 3 levels.
Okay.
And it can't escape, and we have to guarantee to NASA that they won't escape.
Well, I know it's been exciting for me to be involved in this. It's been one of the highlights of my career to help out with these experiments, and to know that something I helped on is actually being tested as an experiment on the International Space Station is not something that I would have expected would happen in my career. Well, this has been great, Stefanie. Really enjoyed it.
Yeah, I did too.
Yeah, thanks so much. Thanks for joining us for a Skirts in Science conversation. We're hoping to have a lot more conversations with other women who are involved in science and have science careers.